So I thought, here's my plan. I got too much in this already if we don't get to the very end things. But I'll start by trying to motivate why do we care about power, what are the very basics of power, which probably people are familiar with outside of the neuroimaging setting, and then run through kind of the couple options we have in neuroimaging, several options that we have, and discuss their applicability. Happy to keep this very informal. Please ask questions as we go along. So I'd like to start with why should we care about power? What does it matter? And I think the starting place for this is this paper by John Ioannidis, which at this point has been cited over 6,000 times and has this really provocative title: Why are most research findings false? Or why most published research findings are false? And I think it's useful to walk through the details of his argument because I think it's quite subtle and maybe if you haven't seen this, you should. Everyone should have a grasp of this paper, so when it comes up in a party, you can nod your head knowingly. But let's actually work through what it's doing. The entire argument is based on a positive predictive value, but the sampling units are something strange. So usually when we think about positive predictive value, you go into the hospital, you get a test, the unit is you, a person, right? But now here, the sampling units are research hypotheses. Completely different from how we normally think about it. So you might think about, what is a hypothesis set in cognitive decline of aging? Well, vitamin D reduces the risk of cognitive decline in aging. Exercise reduces the risk. Fish oil, there's a whole population of research hypotheses that we can consider, and every time you read an article, you consider you're kind of like reaching into this population and drawing. And then the question is, given that the finding is positive, which it usually is, how should your belief change about that hypothesis, that particular hypothesis that you're looking at? And that's what we mean by a positive predictive value. Once I see something is true, how should my beliefs change about the true unknown state of that hypothesis? So let's just nail down some notation. So we have a true research hypothesis, we have false ones. Given that the literature is mainly just this row here, that's all I've populated here. So power, we write 1 minus beta. That is the probability that that we get a true finding, given that the research hypothesis is true. And then false positive rates are when we get a positive finding, but actually it was false. Okay. The one thing that this logic depends on is representing this in terms of an odds. So this is the probability of a true research hypothesis to the probability of a negative one. That's the odds of a true hypothesis. And you can go backwards and forwards between probabilities and odds. So positive predictive value can be worked out by simple application of Bayes' theorem, which is this is the probability of saying, now that I have seen one paper that has a positive assertion about a research hypothesis, what is the probability? What is my belief now in that true research hypothesis that this paper is trying to reflect upon? And actually has a relatively simple form. It's the power times the study odds over the power times study odds plus the false positive rate. So it just depends on these three things: power, your prior odds for this given type of research hypothesis, and the false positive rate. So what was the title of the paper? It was Why Most Research Hypotheses Are False. So he's making a fundamental argument about when is the positive predictive value greater than 1/2, and how often is it less than 1/2. So you can do this, and you can solve for, okay, when is this greater than 1/2? Well, actually it turns out this is equivalent to this statement here. So power times prior odds greater than alpha. If you had some master's level statistics, you might know that the test power can never be less than this false positive rate. So 1 minus beta is always at least alpha. So you could say, well, what's there to do here? So this is saying that the power is always going to be greater than alpha. And then what are these study odds? So if the study odds are 1, there's nothing to see here. Your belief is going to be, basically your positive predictive value is always going to be greater than 1/2. So most research findings will be 1/2 if you believe the prior odds on your study hypothesis is 1. The whole exercise here is actually seeing how these things map out over variations in R. The other thing to note is that why do we care about positive predictive value, and why am I talking about this now and I'm talking about power, is that you can show that this expression, the positive predictive value, is roughly the power. So if you want to know how much power do I have, what is the positive predictive value of a given study, the first thing you should ask was, what is the power for this study? So that's the main sort of driving thing here, is that power is related to the positive predictive power. If you have bigger positive predictive value, you have better power, worse power, lower PPV. And this is the main finding of Ioannidis' paper, which says, okay, if you live over here and you really believe that every single research hypothesis is true, then yes, if you have a well-powered study, then this is, you're up here. Just ignore these, I didn't go into this. This is actually worse things that can basically corrupt the quality of the studies. But we'll just stick to the blue line here. And then as the prior study odds go down, so you might think, so around here are the prior odds for studies of extrasensory perception, right? So that's like, even though it's a well-powered study, even if you believe there's an effect, you might have high skepticism that those research hypotheses are true. And so you belong to be down here. But you can see that there are a lot of settings here, and again, I didn't go into it. These are things that basically affect the quality of a study, can drive this below a half. And certainly, if we're in low power, then most of these settings are below a half. So this is the crux of his argument, that if you actually acknowledge that not all research hypotheses are true, that is the true state of them, the little h, then your positive predictive value is going to be, can be quite small and low, especially if the power is low. And you might say, well, most people, have good power. Well, do they? The other piece of evidence here is this study from Button et al., which also John Ioannidis contributed to. And this is a neat little exercise. They did a meta-analysis of meta-analyses of papers in neuroscience. And they excluded imaging because they didn't know what to do with imaging. So this is just they scoured the literature for meta-analyses, and they collected them all, and then did this cute trick. In general, we do not know what is the true power or what is the true effect size for any given study. You don't know. That's why you're conducting the study. But one thing you could reasonably do is say, if your study has made its way into a meta-analysis, why don't we pool all the effect sizes in that meta-analysis? Because now you have a lot of data. You can estimate that pretty well. And let's assume that the true unknown effect size in each study came from that meta-analysis. So it's a cute way of actually taking a study in the literature. So there were in a total of, I don't have it here. There's something like, I think 600 studies that we now can go back and say, okay, now I know what the true effect size is for the study. I know the N. I know what alpha level you have. I can now infer what would be the power in each of these studies, even if the people hadn't run a power analysis. And this is a depressing thing. They got this histogram, which is also depressing because they used Excel to make this plot. But you can tell that the median here is 21%. So that's saying half of the studies had a power of 20% or less. And this little spike here, these are all water maze experiments with rats. There was something special about these. They were kind of like very obvious experiments in some way. So this is really depressing. So whether you go by the positive predictive arguments of sampling a population of research hypotheses, or let's say that actually all these research hypotheses are true, and the power is 0.2. Well, what does power mean? Power is saying, if there is a true effect, what's my probability that I will get a positive effect? That's saying 4 out of 5 times that you replicate that experiment with the same N, same settings, you'll get a negative result. And only 1 out of 5 times where you get a positive result. That's what it means to have 20% power. So I'm trying to getting ahead of myself, but this is all to motivate why should we care about power. We should make some attempt to understand the power of our analyses. So let's go backwards and start back to basics. Where it is easy to define power and set things up is for one test. So power is a probability of rejecting the null hypothesis when it is false. In order to determine power, you have to set some things. You have to set the sample size, you have to set the alpha level, and you need to set the variability of the effect and how big is the effect, or you can combine these two into an effect size. So how does this work? So you have a null distribution of your test statistic, you have an alternative distribution, and we use the null hypothesis to decide whether when we actually conduct the test, whether we're going to reject the null hypothesis or not, and we calibrate it by making sure that this area here is alpha, say 0.05, and then we can tally up that area as the power. So that's power. One thing when you're actually doing this in practice is that if we're going to do power, you generally have to think about it in the units of the analysis. So for example, whereas if we can conduct a t-test, we usually think about what is a big t-test? 2, 1.96, like that's a big value of t. Well, you can re-express that in terms of the sample mean as well. So instead of saying, I'm going to reject the null hypothesis if my t-statistic is large, you also can say, I'm going to reject the null hypothesis if my sample mean is larger than the critical value times standard deviation divided by the square root of n in this one sample case. So I can equivalently be thinking about this thing here in t-statistic units, or I can actually be thinking in absolute units, which may be more useful. So this is, sorry. This is equivalent to a one-sided confidence interval. For simple univariate tests, there's a duality between testing and confidence intervals. That's right. So just for the concrete example, if I had 10 subjects and I somehow know that my bold standard deviation is 0.5, and I could consider different levels of bold, true bold effect, 0.1%, 0.2.5. If you're not familiar, if you don't have this intuition, you should know that a 1% bold effect is pretty solid, something that you might expect only to see in primary sensory, and you'll never get, I mean, I think you need only in primary visual or primary motor would you ever see up to 5%. So interesting cognitive effects are in this range, 0.01, 0.2, 0.5. I mean, a lot of papers I've seen, it's like never better than 0.1%. So that's what we're living with. And so this is what the power plots are looking like. I can do this all in terms of the raw effects percent change. So here is a 0.2% change, here's 0.5. And you can see that, of course, to the bigger percent change, we have more power. So the first thing that you have to appreciate with power is that it's all a speculative exercise because, of course, before you collect your data, you don't know what these values are. So this is the hardest thing in any power analysis outside of neuroimaging. And it's also why in a lot of cases, a lot of statisticians don't want to sully themselves with power analysis because it's sort of detached from data. It is an exercise that is really all about sort of making guesses. And it's something that a lot of people sort of don't want to mess with. Okay. So the one thing I'll put on the side, too, is that one thing that you have to appreciate is that power analyses are predictions about future analyses. So while we may collect some data to help us learn about those deltas and those sigmas, you cannot make useful power statements about a given data set that you've collected. I know this is hard, but again, I haven't even started talking about imaging yet. But just for traditional, good old-fashioned, I have one hypothesis, I have one test statistic. Now I want to see, what was the power for that analysis, what we call post hoc power? Well, so either the null is true or it's not. If the null is true, you have 0 power. If the null is false, and you set the true effect size equal to the data that you observe, you'll always have at least 50% power. You can never have less than 50% power when you conduct a power analysis based on the data at hand. So in general, if you sort of read up on this, people say, danger, 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 do not do post hoc power. Using the very same data to estimate a power is, it'll always be great. It will never be less than a half. So it's something that it's dangerous, and I wouldn't say dangerous. People get too worked up about it, but just say that there are caveats, and it's something you can say to make predictions about future data sets, but it doesn't really help you interpret what's going on in a single present data set. Okay, so now I'm going to actually start to roll through a bunch of, try to deliver on the title, practical power strategies for neuroimaging. And the first one really is basically to pretend like you're doing a clinical trial. And because here's the problem. If we're really trying to say that we're doing power for neuroimaging, so first of all, you might have to deal with multiple testing, but really that's not the hard part, because you do some multiple testing correction, or maybe use some historical data. Random field theory usually gives me a threshold around 5. Okay, that's fine. That just sets the alpha. That's just one parameter that goes into the power analysis. So that's the least of our worries. The problem is, what is the alternative? If you're trying to pose this voxel-wise and saying, I want to set the power for my voxel-wise analysis, and you have 100,000 voxels strictly, you really should be setting the delta, the capital delta divided by sigma for every single location in the brain. And that's just a huge space. I mean, how could we ever reasonably, make guesses about what that is? So, don't forget. So it's the deltas. You need the capital deltas and the sigmas too. So it's just crazy. So one practical way forward is this clinical trials approach, which is basically saying, I have one primary outcome that I want to power my analysis for. And this is sort of the approach I've recommended over the years for people, especially when you're trying to get ethics approved. I'm not sure how it is here in the States. You always have to supply some sort of formal power analysis to justify the N number of subjects that you're recruiting. And this is the technique that we invariably use, just to say, I want to ensure that no matter what, when I'm analyzing my data in this very focused, simple way, I will have sufficient power to detect an effect. And so I'll define my outcome here. For example, I want to know the average % fold change in the amygdala. I'll need some previous data to estimate the delta in the sigma. Previous literature. I put down pilot data here. That the one thing I'm cautious about, pilot usually means single digit N. And one of the problems here is that estimating a mean is hard enough with small data, but estimating a standard deviation is just catastrophic when you have very small amounts of data. I mean, I wouldn't really trust a standard deviation with fewer than, say, 20 subjects or something. So this is one of the problems. So I say pilot data, but it's really hard to trust the standard deviations that come out of the really, really tiny subjects. So ideally, you have some studies in the lab you can pull together to get some guess. And then once you have the delta and you have the sigma, you of course need to set the alpha. Now you might have multiple regions. Maybe you're going to look at left and right amygdala, or you've got some other regions. Then you can adjust the alpha, say with just a simple Bonferroni approach, dividing by the number of regions of interest, and then go use your favorite power tool. No need to do anything special. So you basically have eliminated the complexity of brain imaging in the final analysis, and that frankly is the most kind of simplistic and practical way. Yes?
Quick question. Can you just comment on the difference between not ever doing retrospective power for a study versus using those same data to do future power calculations?
Well, that's fine, because that's about the future. Yeah.
Computation identical.
Yes, yes. So the question was, what's the difference between prospective power and using a data set to do power? I mean, it is the very same thing. It's a question about, are you making a statement about the data that's in your hand, or are you making a prediction about future data? It's subtle, I know. It's a good point. Okay, so that's the simple sort of approach that kind of gets rid of the complexity of brain imaging. I don't think I stressed it here, so let me just mention it. That the other thing that makes this challenging is when I say previous literature, I'm hiding a whole bit of sort of pitfalls there, because most of the literature is filled with tables of XYZ coordinates. And the problem with those, well, first of all, they may not always report a Z or a T value, but when they do, those are the values at the peak. They're actually biased by two effects. They're biased because they're the ones that actually got above the threshold and made it into the table, and they're also biased because they are peaks. They are local maxima that have a little extra juice to them. So this is a challenge. So when I say previous literature, this assumes that you have ROI or unbiased estimates. I don't have the slide up here, but if you recall, there was a kerfuffle with voodoo correlations, and that was an entire paper about that was based on a meta-analysis that showed there was a distinct difference in the estimated correlations between behavior and brain responses, depending on whether they just looked at the peak in the statistic map versus used an independent location to read off what the effect size was. Okay, so even this has some complexities. Yeah.
So on the previous slide, so I understand what you would do if you had, say, if you were using an anatomical mask in the amygdala, and previous literature had also used that. That seems like the best case scenario. But what if you were using someone else's peak coordinate, as one often does? And can you then use that peak coordinate estimate?
Sure. The key thing is that the location is independent of the data that you have at hand.
Right.
So that just makes sure that there's no circularity.
For the power analysis.
No, I understand. But if you have your own data And you can't just go to the peak because that has a circularity bias. But if you go to the literature or to a friend's data set, oh, they got a peak at this XYZ location, you can go look into your data at that XYZ location, and that's fine. That will give you these parameters to do a power analysis about some future data.
Right.
Good. So just to stress why this is not the end of the story, it's not so flexible. So that is going to tell you about the power for a particular design and also for a particular sort of number of runs, particular configuration. Because if you perturb that design really at all, you no longer can sort of say that that's applicable, because this is really just a statement about that particular design, that whether it's event-related versus block, how many different conditions. It's really just a statement about that. So if you change it in any way, the power will be different. And of course, there's also the fact that of course, it is not a spatial method. You've kind of dumbed it down, so it is just a set of numbers, and it is not doing anything spatial. So the next methods I'll show will try to address this and with increasing degrees of complexity. So to successive students of mine, I've sort of told them, the adage went in the movie The Graduate, plastics. I used to tell my grad students, power. It's like green fields. No one's working on it. So one of my first graduate students, Jeanette Mumford, picked up this area. After we read the Desmond and Glover papers, so this was kind of one of the first sort of careful studies of fMRI power that kind of looked at, what do we really have? How does power vary with different factors? But they assumed a simple boxcar model. It did not do anything sophisticated. Well, basically ignored the temporal correlation, and it did not have a very sophisticated approach to deal with the group, the within versus the between random subject variation. So we took on the project to do basically that work again, but allow for arbitrary experimental design, account for autocorrelation, and specifically account for the intra and inter subject variation separately. So this is a cartoon of how we all analyze fMRI data at the task fMRI data at the group level. We do it in a two-staged approach. At the first level, we fit a general linear model that expresses the experimental design, and that gives us parameter estimates for the relative height. We always summarize those parameter estimates with some sort of contrast. Usually we're just boiling this down to a single number. And then that gives us an estimate, and then we feed that up to a second level model where we now have one observation per subject. So this is one observation per time point, and this is a group level model of one observation per subject. And then this might be a group model, say, patients controls, males versus females, what have you. And then we're interested in the group difference, and so again we use a contrast and we get an estimate here. So the thing that's going on behind the scenes here is there are a lot of things, if we're going to specify power, that we need to have to determine. So there's actually how noisy is the first level data, how much between subject variability is there. Also at the first level, there is temporal autocorrelation. And I've masked that here by just this is a square n by n matrix V is one of the parameters that would have to be considered. And then also here the sigma G, which is what is the between subject variation in this response. So there's a lot here. So if we're going to do a full comprehensive power analysis for group fMRI, accounting for all these things, we need to account for the following. We need to say we've got a contrast of interest that has a specific magnitude, and we have to know what its non-centrality parameter. So what does this depend on? Well, it depends on the effect size, but also it depends on the design at the group level and this variance at the group level, which in turn depends on all this. So this is just getting an idea at the complexity. So the things that we know or can set are the number of subjects, the alpha level. Again, if we're correcting for a number of regions or voxel-wise, we can set that accordingly. We have to know what contrast of a group model is. So for example, this would be 1 minus 1, and the design that you saw on the previous page for indicators for group 1 versus group 2. And then at the first level, we also have to set the design matrix and contrast for each subject. Now, I guess we can make lives a little bit easier for ourselves and assume this is the same for all subjects, but still, we have left all these things which we now have to guess. So we have to guess what is the effect magnitude at the population level, what is the between subject variability in the noise-free estimates in each subject. And of course, they're not noise-free, and they're characterized both by a noise magnitude and, holy camoly, this n by n noise correlation matrix. So I don't know about you, but I don't have much intuition on how to form this n by n correlation matrix, just, de novo. So this is one of the first things that we had to grapple with is, this is huge, right? There are different approaches. So SPM, for example, does use a simplifying approach that uses an approximate AR1 model. FSL uses something that's more complicated, which is good. I like FSL's model, but then if I'm doing a power analysis, how am I going to specify this? And I'm going to write it down in a paper. Here are the 17 numbers that made up the autocorrelation function at a given voxel. This is more flexible, but then has more parameters. And if you're going to do a detailed power analysis, you need to tell people what it is that you use. So we decided on using this simplification. Let's assume that the autocorrelation in our fMRI data at the first level have this form, an AR1 plus white noise model. And then we just need these three parameters to describe it. What's the magnitude of the white noise variance? What's the magnitude of the colored variance? And how strong is the serial correlation in time? So that goes from, oh my goodness, how do we specify this n by n correlation matrix to, we will say that it is fully determined by these three numbers, which I can conveniently report in a paper. So I'll just sort of work through this as to what this looks like for a block design with TR 3 seconds, standard HRF. And I'm going to use, I have to come up with these parameters somehow, so I'll estimate them from a study. This is a functional imaging analysis contest data, which was sort of just released at the time she was doing this work when she did her thesis. And this is looking at the same different speaker effect, I think is what it is. Okay, so what's neat, though, is that once you've specified all these parameters, you can then change the dials on this. So I can change my XK, the within subject design. I can make it shorter, I can make it longer. I can have more repetitions of the blocks or fewer. I can change the number of subjects given that I've set these parameters. So I used the data for this sentence comprehension effect, and it told me that there was basically a 0.7% bold effect. The between subject variability of that was 0.4 standard deviations in percent bold change units. The autocorrelation parameters was 0.7 with AR noise being 0.1 and the white noise being 1.3. And we're going to say that this is saying correcting for, say, 0.05, say that would be for 10 regions of interest, be the correction. So this is fun. So now I can actually change this. I can change the number of subjects, but then I can also change the number of on-off cycles I did on a block design. And you can see that for any given number of subjects, the power increases with the number of cycles. But you are limited, right? So if you only have 17 subjects, you're just never going to quite get to that power. Whereas if you have fewer subjects, if you have more subjects, you can get by with fewer on-off cycles. So this is the advantage of this comprehensive approach to power. We can actually play with these things. I can change the inter-subject design, in this case changing the length of the runs, and I can change the n, and I can play with these things. You can also do fun little play-offs of like, well, what if I just say I want to minimize the cost expenditure to get to a given level of power? You could do that in total scanner time in minutes. And so you can see that if you examine more subjects, you actually are going to use less scanner time, which I think is compelling, except, of course, that it is expensive in human time, right, to get subjects in and out through the scanner and tested and everything. So that's not so. But you could say, well, how about I add a cost per subject? It costs me $300 to do each subject, and let's say it's $10 of each scanner minute, and then you get a global optimum. I mean, we didn't bake it like this. It turned out to be 20, which is everyone's favorite sample size. But just saying, this is the sort of things you can play with once you have a comprehensive model for your power. So again, like any power analysis, this is incredibly dependent on the parameters of the true state of nature of the effect size, and now, and for us, both the between subject variance and the within is parameterized by this particular AR plus white noise model. But given those parameters, I can play lots of games and look at lots of trade-offs. Okay, so the sort of the limitations to this approach is that it does ask more of you. You need to know these parameters, the variance of the AR, the color noise, the independent noise, and the AR coefficient, and the between subject variance. Fortunately, there are tools to estimate this, but again, you're going to be hard pressed to sort of just come up with these values because we don't routinely present any of them. We don't, as it turns out, AR plus white noise is not a standard model. It's possible we could fit it and extract it from other things, but I mean, we do. I'll show you on the next slide. But it's not something that I particularly can roll off some intuition of like, well, the last 10 studies I read had this kind of AR white noise, this kind of AR row. So what is really important is that Jeanette has implemented and has continued to maintain this tool, fMRI power. So for completed group analysis, you can enter this, whether it's an FSL or SPM, and it will go and estimate those parameters that you need those 4 parameters that you need, and then it will then tell you what the power is for a different set of ROIs. So that's really handy, and so that's kind of the most comprehensive. And if you really want to play around, with different trade-offs, this is, I think, sort of the best approach. So, any quick questions on that before I move on to the next one? Okay. So I want to give a shout out to the other work that came from a former student of mine, which is Satoru Hayasaka, who tried to address the other aspect, which is the work that I just presented, while it's very sophisticated in terms of the temporal modeling, makes no attempt to do anything fancy in terms of the spatial modeling. And so after he graduated and was an independent researcher at Wake Forest University, and he's now at the University of Texas at Austin, Satoru Hayasaka developed this method for doing cluster-wise inference, for determining the power of cluster-wise inference on the basis of random field theory. So what you may not appreciate is that random field theory, which gives us our p-values in SPM and FSL, is actually entirely based on the null hypothesis. It's beautiful mathematical theory for mean 0, variance 1 fields. And so he worked out the results for what happens if you don't have a central field, but a non-central field. And of course, not everywhere, but just say, for example, in some pre-specified regions. So he worked this out, and so you basically, whereas the normal results for the maximum distribution look like this, he was able to work out what are those maximum distributions when you have a shift, and in particular for the size of clusters. So he's able to, again, load up data from which you then can estimate the random field theory parameters and then work out the power for a given size. You'll see this has not really been maintained. I'm not sure if it functions with sort of current versions of the software. And as you might know, I have a lot of reservations about random field theory, especially cluster size inference and its accuracy. So I'm not so enthusiastic about this one, but just to say this addresses a shortcoming of the previous work, and that it actually tries to account for the spatial nature of the data and the fact that oftentimes the data are smoothed, and you could actually use this to give you maps of effect size for detecting a cluster of a given size. Okay, so move on to the next one, which I think is another practical approach that does deal with the spatial aspect. So I was introduced to a student, Joke Durnez, who at the time was at the University of Ghent in Belgium, and she wanted to look at power, and she too tried to start with cluster inference power, and she way long ago, way before the cluster failure paper, noted and discovered on her own that the random field theory results for cluster size were just a mess. They did not work with any level of accuracy. And she's so frustrated by that, she almost gave up, but then she thought, oh, wait a minute, there's one other thing that random field theory delivers for us. It's the inference on the peaks. So you might know that in SPM, the table of the results no longer says voxel-wise inference, it says peak inference. So it turns out it makes no difference for the family-wise error corrected values, but for the false discovery rate values, those are actually peak false discovery rates if you haven't turned off topological FDR. And so that we actually have uncorrected p-values for each of these peaks. Confusingly, the uncorrected p-values in the SPM table are not peak p-values, they are the regular univariate p-values. We could add another column and add the uncorrected peak p-values, but that's another story. But so this was, she observed that the peak p-values from random field theory were actually fairly accurate. And why don't we see if we can use them to make an approach to estimating power. And for this, what we'll have to do is we'll have to estimate how big is the non-centrality parameter, the Cohen's d, how big is the region, and we'll also have to know the smoothness because we're working with random field theory. So, I'm sorry, that's just the story I just told you, that she tried all these things and we ended up with peak-wise. I should also say, yeah, it would be nice to do voxel-wise, but also to do voxel-wise power, accounting for the spatial aspects, would require a detailed model of the spatial correlation, which again, we have a working model of the spatial correlation that's using random field theory, but we know it's not accurate. Again, that's something we learned in the cluster failure work. The nice thing that's beautiful about peak results is that the peaks are roughly independent. So even though that we use knowledge of the smoothness to figure out what their distribution should be, each individual peak can be treated as an independent random variable, and that really simplifies the analysis. So what do we need to specify? So now we have to specify what proportion of the brain is showing an effect. We'll call that pi1. And in those regions where there is an effect, what is the response magnitude in a Cohen's d or delta? Sorry, I've changed notation there. And of course, the other usual stuff, alpha and the number of subjects of n. So again, now from pilot data, the same thing. But the thing that's nice here is that unlike Jeanette's work, which needs the full group analysis, both the first level and the second level data, this needs but one statistic map. And this really impressed me with what she did because she used methods that are out there to estimate these parameters from the data. So this is a distribution of the p-values of the peaks. So this, in a typical brain image, we have lots of peaks. And so this is nice. So we can actually look at their distribution and infer, well, actually, it looks like there's a baseline of uniform p-values. So something you may not know is that under the null hypothesis, p-values are uniformly distributed. So evenly spread between 0 and 1. And look at that. That's this flat bit here. And there are a number of different ways you can use to model empirical distributions of p-values that will tell you, I think this much of it is null, and I think this much of it is non-null, using some distribution like a beta distribution or what have you. So the approach that we found that worked the best was to say, on this p-value distribution, we'll estimate pi naught. So here, this particular example, 45%, a lot of the brain showed activation. So this is saying 45% of the brain is activated based on this. And then we did a mixture model. So in some ways, this is the very same data. These are now the peak heights. So these are the peak heights, just the statistic values. There's a one-to-one mapping between the peak heights and the peak p-values. But we found it was easier to do this in two stages. Stage 1, estimate this pi1, what proportion of the image is non-null, fix that value, and then go over here and do the mixture model to figure out, okay, I know the null distribution of peaks, it's an exponential distribution, a shifted exponential. What is the mean of the non-null peaks? Because that's the other thing I need to know. I need to know the Cohen's d of where there is a signal, how big is that signal? So in this two-step process, we're actually able to estimate these two things. We estimate the proportion of the nulls, and then we also can estimate what is the peak distribution. And here we get both a mean and a standard deviation to characterize the non-null peaks. And then given those sets of values, we can go and then do power analyses on future data sets, again, assuming that we have the same proportion of the brain activated and we have the same distribution of alternative peaks. And you can try, of course, you can use different measures for multiple testing. You can say Bonferroni. There is a way to actually use Benjamin and Hochberg in this setting, or you might use random field theory if you, again, because we know what the smoothness is or uncorrected. And with all these things, you can see. So if I want to have evidence that is sufficient for family-wise error, that's telling me I need to be out here at 41 or 42 subjects. If I'm willing to accept a false discovery rate measure for controlling multiple testing, then it's sufficient to do 27 subjects. And so again, what makes this work, I think, important is that there is a practical implementation that Yoka has developed. She went to work with Russ Poldrack at Stanford. She's now actually working for a nonprofit, but she still continues to maintain this. This is a R Shiny app. You upload a statistic image, and it will go and will fit these non-null distributions, and then allow you to do your predictions of power based on that. So it's been used some. I will say that if you have very, very rare, very, very spare signals, so if you have a very, very tiny proportion of the brain that's activated, it may be hard to estimate that in this setting. But if you have, as long as these two different pieces can work, estimating the proportion of the brain that's non-null, and then estimating how big is the non-null set of peaks, it actually works great. Okay, how are we doing? So I think I have time to talk about, so those are the practical power strategies. So one is get rid of the imaging aspect. Use some independent way to identify a location in your data and estimate an effect size, and then do a power analysis from there. Or if you're lucky, search through the literature to find some effect sizes that are not just based on peaks. Or use a detailed method where we actually have to estimate some aspects of the temporal autocorrelation and the between and the within subject variance, or Yoka's approach of just using aspects of the peaks. So those are my three offers, and I'm sort of downgrading Saturo's work because, again, we've learned more about cluster size inference and random field theory. So I'd say those are the three sort of practical approaches. In the last sort of 5-10 minutes then, I just thought I'd sort of tell you about some related things that I think are of interest, and I think are sort of in the same theme of sort of open science and sort of meta science, if you will. So this is some work that you may have related to meta-analysis on something called the file drawer problem. So file drawer problem are studies that have been conducted and then never published. And I've done work with both intensity-based meta-analysis, where you actually have the images, and the coordinate-based meta-analysis. And what we realized is that this coordinate-based data, they all take the form of peaks. So here's the points from one study. This actually, I think, had some 40-some peaks in it. And this is from a whole bunch of studies, 164. And it turns out that count data obeys laws of statistics. There are just a certain number of ways that count data behave. It's always 0 or greater. They're integers. And there are just a small number of distributions which describe count data. And so we had the idea of like, well, from the brain map data, I can get 13,000 experiments from which to learn what are the behaviors of these counts, and why is that interesting? Well, so here's their distribution. It kind of looks like maybe an exponential, or maybe it could be Poisson, who knows? But here's the neat thing. There's one thing distinctive about this distribution. There are no 0 counts. A 0 count statistic map is one which is a null response, which is a negative result, right? So our idea was, can we use the natural law of count statistics to infer what should be the 0 count of these? Given that there is randomness in how all these statistic maps are created, and some of them come out to be 0, and what happens to them? We don't see them. They don't get published. Can we use the characteristics of this distribution to infer what that is? So we did. I learned about some distributions I never knew existed. So the one thing, Poisson counts do a horrible job. These counts are not Poisson distributed at all. If you do a negative binomial distribution, that does a much better fit, and there are even more esoteric distributions you can look at. But the key thing is, it turns out there's standard software available in R for fitting count data that is truncated, because we don't want to say that the data we're seeing could possibly be 1. No, by construction, could possibly be 0. No, by construction, the data can never be 0. We're going to fit these models conditional on the data being 1 or greater, the counts being 1 or greater, and then using those sets of parameters to infer what would be the rate of 0 count experiments if we could observe them. And so we can do that, and this is the fit we get from the negative binomial. Also, we can, because the general regression framework that we're using, you actually can include things like publication year, study domain, classification, what have you. And when we do this, and we express it, sorry, I should go back, this is important. So the thing that we can estimate is if zeros were possible, we can renormalize things and say, what is the probability of observing a zero? But I think that's not so interesting. I think what was more interesting is, what is the zero count prevalence of studies per 100 studies? So for every 100 studies I observe, how many zero count studies are there out there that I did not observe? And that's a simple transformation of this probability of y being zero. So the rest of the results I'll show you are all about PZ. Rate per 100 of studies that should be out there, but are not because we haven't seen them. And so we did look at this as a regression in terms of publication year, also in terms of number of subjects per study. And this is what we see. Kind of no matter how we do it, it's all usually around 10. And in the write-up, we've tried lots of different things. We've tried lots of different models. And it varies some, but it never goes below, say, 6 or 7. So that's basically saying there's, at least 5 studies that have zero counts for every 100 that you see in literature, which is maybe not surprising. And when I've shown this to people, some say, well, it's surely higher, right? Or the other thing, too, is that I cannot say anything about the quality of these studies. These could be junk studies. I mean, these could have horrible circularities. They could be horrible methodology behind them, and there are actually lots of false positives. I'm just talking about the random characteristics of all the studies in the literature. And given those, how many zero ones should be out there. And the other thing, to be fair, of course, is that the brain map database has no capacity for entering zero count studies. So it is possible that some studies say, oh yeah, and we looked at blah, and we didn't find anything. Because the brain map curation is based on finding tables of XYZ coordinates, and no one makes a table that's empty, right? Yes?
Could you comment on perhaps might be a publication bias? If an actual study truly had zero, its data gets turned over and over and over until one spot pops up, and then out it goes. It's bad practice. We know it has gone on. Can you sort of see that and anticipate that?
We looked at it for just the count data. Because remember, we're throwing away a lot. We're throwing away space, and all we get is an integer count per study. And we posed some models about trying to say, well, what were things that were zero, and then they got promoted to be non-zero. And I just couldn't convince myself there was enough information in just this count distribution to infer that. I mean, we could fit them, but I had no confidence that we could sort of. I mean, if you did something, if you really committed to one model, I mean, this wouldn't work. But like, let's say I really truly believe that it should be Poisson distributed, and then you work out what is the mechanism that messes up the data to give the weird distribution that we do get. I mean, that would be one approach. But you have to start from some strong beliefs about what good data looks like. Okay, and then this varies with different domains. We looked at normal mapping, which is just no diseases, linguistic effects, disease effects. They're all sort of were between 5 and 10% for this rate. So yeah, so it's kind of I'm not sure what it means. We all have worries about the quality of brain imaging data, and so I'm not sure how this changes our knowledge of this, but it is just answering the question of, is there a file drawer effect in brain imaging? Like, look, probably, yeah, and this is the rough magnitude of it. I think I've already made some of this discussion points here. Yeah, so I think I'll just skip on that. So we've submitted this, and the reviewers hated it. So went to Neuroimage, and now we're trying in neuroinformatics. It was not a pleasant exercise. So it's on archive, but it's still in revision. And I think I'll just do a shout out to the Kobe-DAS. So Johan mentioned this effort, this thing I did for the Organization for Human Brain Mapping. When you are sort of writing your first paper and you're kind of thinking, am I including everything I should? The purpose of the Kobe-DAS exercise was to say, can we get a bunch of experts around to sit down and agree on what should be reported? We actually started to say, let's tell everyone how to do their experiments. And we realized that you can't do that. There's just so many different things that are done, too many different types of experiments, too many different ways you could possibly analyze your data. We really could not be prescriptive. So this is a big, long sort of exercise in saying, how should everything be reported in your analysis and in your study. Everything from how you did the design, how did you do the data acquisition, how did you do pre-processing, modeling, and then this is getting more prescriptive, so the modeling and inference has a little bit of prescription. Data sharing, this is early days, sort of selling suggestions on how you could do that and things to maximize reproducibility. But especially these first 4, I'd say, are basically saying, if you're gonna try to produce a publication that someone else is able to pick up and actually conduct that experiment again, what do you want to make sure is in there? And it's based on these kind of really boring, sort of exhaustive tables of, things that most people probably wouldn't. Do you want to report the number of subjects you approached? I mean, probably not in a lot of cases, but if you want to be really exhaustive, you put that number of subjects consented, people refused to participate. It's really kind of a thought process. And the things that are yes are just kind of everyone really can agree, we really, really should include these. So I'll just point, it's a resource that's out there. I think it's especially when if you're trying to do training or if you're new and you're writing up your first paper, just some things to think about to make sure that you've sort of included everything that describes what you did and maximize the chance that someone else could reproduce your work. And so then from there, I'm just going to skip to the end because this just gets, that's the conclusions, yes. So why do we care about power? Because power is intimately related to this positive predictive value. Why should someone believe your results when you look at the study? So if you want to believe your own results, you want to know that you actually have high power. I mean, in some ways, it's kind of self-evident. If there truly is no effect, you can only be staring at false positives to, I'm really, really sure the effect is there, and then so everything that you could possibly see is true positives. And the power is just trying to tell you where you are in that spectrum. I presented a number of methods, the number of whole image methods, both the fMRI power and power map, but as a kind of the most simple basic approach, there's nothing wrong with the targeted outcome if you have the data and effect size on hand to make that prediction. So I'll stop there. Thanks for your attention.